This video describes a procedure used to induce toxin synthesis in Fusarium species, in this case Fusarium graminearum, and the protocol for whole protein extraction used for proteomic studies in our laboratory. The length of the procedure is 16 days, 4 days for fungal growth, 10 days for toxin synthesis, and 2 days for protein extraction. After 4 days, colonies are still actively growing toward the edge of the plate. Typical aerial mycelium is formed and is collected using a sterile blade under a laminar flow box by gentle scratching the surface of the plate. Mycelium is cut into 5 small pieces and added to an Erlenmeyer flask containing 25 ml of toxin-inducing media. Toxin induction is more efficient in dark, thus the flask is covered in aluminium. Cultures are then shaken for 10 days at 150 rounds per minute at 25 centigrade. The medium is of acid pH and contains high concentrated saccharose solution that, together with darkness, is known to facilitate toxin synthesis in Fusaria. The mycelium is then separated from the toxin-containing liquid by filtering on the Myra-closed tissue. The liquid can be used to measure toxin content, while the mycelium is used for protein extraction. To avoid media carryover, mycelium is washed 3 times with sterile water. The excess water should be eliminated. Afterwards, liquid nitrogen is added and samples can be stored at -80 or used for protein extraction. The protein extraction procedure is based on the use of SDS and TCA and consists of different lysis steps. Here we propose a method that encompasses multiple operators to carry out at the same time the extraction of biological replicates. A different operator carries out each replicate. This procedure takes into consideration differences in the extraction procedure that can be generated by different operators who are processing the replicates. It should therefore assure higher robustness when biological replicates are compared, as the operator variable is included in the replicate. At the same time, the use of multiple operators reduces the time of processing. The sample is ground in a sterile mortar using liquid nitrogen until mycelium is a fine powder. This step is crucial for the quality and amount of proteins. Mycelium is collected in 10 ml Teflon tubes in a proportion of 4/10 of the total volume of the tubes. Lysis buffer containing SDS and different protease inhibitors is then added. The tubes are then shaken until a homogeneous solution is obtained. Then the samples are shaken for 30 minutes in the cold chamber to be further homogenized. Samples are boiled in order to dissolve cell walls and hydrophobic proteins. The presence of SDS prevents the formation of oligomers that abort precipitation of proteins. A centrifugation step separates 3 phases. Proteins are suspended in the transparent solution that is collected in a new tube kept on ice. The pellet is then used for performing a second lysis step, repeating the vortexing, boiling and centrifugation. The supernatants from the two lysis steps are combined in order to perform precipitation with glacial precipitation buffer containing acetone, trichloroacetic acid and DTT. The samples are then stored overnight for 16 hours at -20 centigrade to allow protein precipitation. Proteins are situated at the bottom of the tubes. To improve precipitation, tubes are centrifuged at high speed for 45 minutes. The pellet is now well visible. Supernatants can be discarded using a 5 ml pipette. TCA is then removed by adding 25 ml of glacial washing buffer containing acetone and DTT. It is then thoroughly and vigorously shaken. Then a new centrifugation step is performed. The protein pellet is now floating, so attention should be paid to eliminate the supernatant. A second washing step is performed, adding washing buffer, shaking and centrifuging. The supernatant is eliminated and the sample is dried using a speed vac. When the sample is dried, the protein is of powder-like consistency. To store the sample, proteins should be collected from the tube. To diminish the electrostatic interference of plastic, an electrostatic pistol is used and the protein powder is collected using a metal spatula. The proteins are then resuspended directly in the labeling buffer used for 2D Daish. 30 minutes at 800 rounds per minute suffice for solubilizing the protein. Before proceeding to the expensive 2D Daish labeling protein, quality is tested on SDS page 1 dimension gel. As can be seen, the absence of significant smears on the edge of a lane suggests a good quality of the sample.